the last talk this session. Mark Wilber from the University of Tennessee.
All right, great. Well, thank you. And I just want to start off with just a couple acknowledgements for this work. So I want to start off by acknowledging my co-authors that were critical for developing a lot of the ideas that I'm going to be presenting today. And I also want to acknowledge a number of different collaborators from a 2020 Movement and Disease Working Group that was hosted at the University of Minnesota. Obviously, it was all via Zoom. But a lot of ideas that I'm going to talk about today were sort of born in that working group. And I also want to acknowledge a couple funding sources shown here. All right, so let's go ahead and get started. So as we've seen in this session today, there's this ongoing explosion of fine-scale movement data in human systems, livestock systems, and also wildlife systems. So in human systems, the ubiquity of cell phones is allowing us to track fine-scale human movements at spatial scales that we haven't been able to do in the past. In livestock systems, the increased use of proximity sensors allowing us to understand between individual interactions. And in wildlife systems, we've actually been tracking wildlife movement for a long time. But recent advances in GPS technology is allowing us to track animal movements across landscapes at spatial and temporal resolutions we just really haven't been able to do before. And so really the key point here is that these animal movement data are actually currently pretty widely available, but these advances in movement technology are making them increasingly available. Okay, so we have this glut of movement data at our fingertips. How do we process this data? How do we actually make sense of this data? So fortunately, concurrent with these advances in movement technology, we're also seeing really cool advances in the field of movement ecology. I just want to focus on one of those today, and that's continuous time animal movement models. So what do these continuous time animal movement models do? Well, say you have some GPS data. This is a white-tailed deer moving through space. These are GPS fixes. What these continuous time animal movement models allow us to do is, with biological realism, predict continuous time movement trajectories from these animals and predict the inferred uncertainty in these movement trajectories as well. Now, in addition, movement ecology is also allowing us to infer changing behavioral states of animals through time given movement data. So this might be how an animal is changing from foraging to resting through time. All right? And so these advances in movement ecology are really allowing us to understand where an animal is, when it is there, and also what it is doing in that location. And so one of the main goals of this study and that we are interested in is how do we leverage these advances in movement technology and movement ecology to make better predictions about spatial temporal infection risk. And so we did that, and I'm talking about two things that we did throughout this talk. We did that by developing a model that we're referring to as movement-driven modeling of spatio-temporal infection risk. I'm just going to refer to it as Movester for the remainder of this talk. So for the first part of this talk, I want to give you a conceptual idea of what Movester is. And then for the second part of this talk, I want to show you an application of Movester, particularly addressing this question that's come up a few times in this session is, does ignoring fine-scale movements on actual landscapes actually mischaracterize transmission risk? Do we care? Do we need to account for this? All right, so I want to give you a conceptual overview of Movester. Okay, so Movester begins with movement data, and we transform this movement data into a transmission kernel that is epidemiologically grounded. And then from this transmission kernel, we can make predictions about spatial temporal infection risk. All right, so I want to go through each of these components of Movester in turn. Movester begins with movement data, and it can use a wide variety of different types of movement data. It can use proximity logger data, it can use GPS movement data, it can use spatially explicit capture, recapture data. So a lot of different types of data in different systems. For the sake of time today, I'm really just going to focus in on GPS movement data. All right, so this is us going out to some population, say it's wild pigs. We're putting collars on these pigs. We're tracking their movements at, say, 15-minute, 30-minute, hour-long intervals for months, maybe even years, right? Typical sort of GPS study. Now that we have our movement data, we need to transform this movement data into a transmission kernel, an epidemiologically grounded transmission kernel. Right? And these transmission kernels are really the meat or the scaffolding on which Movester is built. All right, so how do we do that? Well, to do that, we have to think about what is a contact. How do we define a contact in terms of movement trajectories? All right, so consider two movement trajectories here. We have host 1 in red and host 2 in blue. How do we define what an epidemiologically relevant contact is given those movement trajectories? Well, the Movester model says we need to consider 5 specific processes. First, we need to think about contact formation. All right, this might be when host 1 and host 2 are within some spatial proximity. All right, that's pretty standard in contact ecology. Second, we need to consider contact duration. So how long are those two hosts within that spatial proximity? Now, to make this contact epidemiologically relevant, we also need to consider two additional rates. We need to consider a pathogen deposition rate or shedding rate and a pathogen acquisition rate. Right? So imagine I'm host 2, I'm the blue host, I'm cruising along in my movement trajectory and I'm infected. I could be depositing pathogen at some rate, maybe I'm coughing, maybe I'm sneezing, maybe I'm defecating. If I'm host 1, I could also be moving along my movement trajectory and I could be acquiring the past or present pathogen that was shed by host 2. Right? So I need to consider both of these rates to understand an epidemiologically relevant contact. Now, because we're thinking about pathogen deposition, whether into the air via a sneeze or onto the ground via defecation, we also need to think about pathogen survival. So if I'm host 1 and I encounter past pathogen shed by host 2, the decay rate and how long ago that host 2 deposited that pathogen matters for the force of infection that I experience upon encountering that location. So the cool thing is, when we take into account these 5 processes, we see that this Movester model can actually encapsulate direct transmission, so when two hosts are in the same place at the same time, and indirect transmission, when two hosts are in the same place but at different times, all within the same framework. And it really recognizes that there's this continuum between direct transmission and indirect transmission, rather than this dichotomy that we oftentimes think about. All right, so let's be a little bit more quantitative about our transmission kernel now. So the exact definition of the transmission kernel is it's the instantaneous contact weight felt by host I at time TA from host J at time TD with units time -2. That's a mouthful. That's totally fine. What a key thing I want you to take away from this is that transmission kernel is defining an individual level, directional contact weight. So how host J experiences a contact weight from I and how host I experiences a contact weight from J, and these are not necessarily symmetrical things. Now we can actually write this down mathematically. So here's one potential form for our transmission kernel. You don't need to worry too much about the various variables in here, but what I want you to pay attention to is that those 5 contact processes are encapsulated in our transmission kernel. We have an acquisition rate. We have a contact formation and duration that depends on our animal movement trajectories. We also have pathogen deposition rates and we have pathogen decay, right? All that is encapsulated in our transmission kernel. Now, the transmission kernel, I'll admit, is a little opaque when you look at it at first, but you can transform this transmission kernel to recover quantities that we're very familiar with in disease ecology. For example, integrating over the transmission kernel yields a force of infection, okay? We have movement data, we have our transmission kernel. How do we now make predictions about spatio-temporal infection risk? There's a lot of different things we can do here, and I do not have time to go into all of them. I definitely recommend you check out our paper if you're interested. We describe some of these different things you can do. Today, I'm just going to focus on one example for the remainder of the time. And that's asking this question, does ignoring fine-scale movements on actual landscapes mischaracterize transmission risk? So to do that, I'm going to use actual movement data from GPS collaring study on 19 pigs that was conducted on a ranch in Florida. And fixes on these pigs were recorded every 30 minutes, and collars were on these pigs for about 4 months. So there's no pathogen data associated with this movement data. But what I'm going to do is I'm going to assume that there's a hypothetical pathogen that arrives in this pig population. What we want to understand is, do the movements, the actual empirical movements of these pigs, how do they influence pathogen spread and invasion on this movement network that we're going to build using Movester? So the pathogen, hypothetical pathogen, that we're going to be using is going to have similar dynamics to African swine fever virus. So what do these data actually look like? So on the left-hand side here, these are our observed data. Colored points are fixes, GPS fixes. Different colors are pigs on the landscape. The sort of gray background is going to be the landscape across which pigs are moving. On the right-hand side here, we use these advances in movement ecology to infer the continuous time movement trajectories of these pigs that we can then feed right into our Movester model. Okay, so we have our data. Now we want to build our transmission kernel. Yeah, now we need to make a few assumptions while we do that. So first assumptions we need to make is regarding contact formation. So in this case, we're going to assume that a contact occurs when pigs are within 10-meter proximity of each other. I just want to point out that all the results I'm going to show following, this 10-meter assumption doesn't matter too much. It could be 5 meters, it could be 1 meter. We get all the same results, all right? Now regarding pathogen acquisition and deposition, I'm going to assume, just for simplicity here, that those rates are constant across animal movement trajectories. Within Movester, we do not have to make that assumption. We can let them vary. I'm just doing that for simplicity here. And finally, we're going to make some assumption about pathogen decay. Here, we're going to assume that a pathogen survives for, on average, about 5 days in the environment. And this is pretty consistent with African swine fever virus. Okay, so we've defined our transmission curve. Now we can want to address this question. Does ignoring these fine scale movements actually matter? We have our data. We have our transmission kernel. What do we do next? Well, this transmission kernel is actually pretty interesting because it defines a dynamic contact network that we can then simplify to a static contact network where nodes are pigs and edges are going to be the force of infection experienced between two individuals. Now, this static contact network actually accounts for a lot of these details about these fine scale movements, but allows for a simple way to sort of explore them. Now, in addition to building this Movester contact network that's accounting for these fine scale movements, I'm also going to do something that we often do in disease ecology. I'm going to build a home range overlap network, right? Just by taking the home range of these different pigs, choose your favorite approach. In this case, the home range overlap network is not going to be accounting for these fine scale movements that are happening within home ranges. And so we can actually then compare these two networks. And the cool thing is the reason we can compare these two networks is that this home range overlap network is actually a special case of the more general Movester model. And so they have the exact same units. So they're directly comparable between these two networks. Okay, so here are these two networks. On the left-hand side is going to be our Movester average contact network. On our right-hand side is our home range overlap network that doesn't account for these fine scale movements. Quickly orienting you on these networks, nodes are pigs. Edges and edge width is going to represent the force of infection felt between two pigs. It's actually pretty symmetrical in this case. So I'm just showing one edge. There's actually in and out degrees on both of these. Size of the node is going to be the betweenness of particular individuals. So roughly the capacity of that individual to facilitate pathogen spread across the network. You're probably staring at these and thinking, wow, they actually look pretty similar. And you're not wrong. They do look pretty similar. But if you actually dive in a little deeper, you see some key differences start emerging. So first off, you notice that the edge widths on the Movester contact network are substantially larger than the edge widths on the home range overlap network. So we're capturing more force of infection. And you also notice that the betweenness values are actually quite different between individuals in these two networks. Now, if we want to encapsulate these differences in, say, one metric, we might want to look at R0 on these two different networks. And when we do this, what we see is that R0 is actually 30 times greater on the Movester network than it is on the home range overlap network. So ignoring these fine scale movements significantly changes our epidemiological inference. So I just want to leave you with 3 key points here. So first off, Movester begins with empirical movement data. And you can use movement data in human systems, livestock systems, or wildlife systems, and you can use many different types of movement data. So we think that makes it pretty widely applicable. Second, Movester generalizes many different types of contact networks. You can use dynamic contact networks, home range overlap networks, even bipartite networks. Those are all special cases of this Movester framework. And finally, Movester is flexible. It's built on this epidemiological foundation, and you can incorporate pretty easily additional ecological or epidemiological complexity. So this could be allowing, say, pathogen deposition to change with behavior over a movement trajectory. All right, so I hope I've somewhat convinced you that Movester provides this epidemiologically grounded way to incorporate fine-scale movement data into our predictions about spatio-temporal infection risk. With that, if we have time, I'd love to take some questions.